Welcome to Bio class. Today we will discuss in cellular respiration electron transport system ETS, electron transport system and oxidative phosphorylation. Okay, so we have completed glycolysis and enters in aerobic respiration with oxygen inside the mitochondrion and the citric acid cycle, tricarboxylic acid cycle also it is done. Now, here it is electron transfer system. This process will start from that the product here, that NADH plus H and FADH2. Okay. So, here the utilization of the energy stored in this. Okay. And also that the oxygen, which is respiratory oxygen we are taking, and it converts into water to neutralize. And this pathway, it will be the electron transport chain that we will see now, and which is called the electron transport system. And then you can see the chemiosmosis and in formation of energy, which is under oxidative phosphorylation. If you recall, then in the photosynthesis also you can see that the formation of energy, that is the photophosphorylation, and here it is oxidative phosphorylation. Let us see this one. It is told that in electron transport system will start, the start point will be that the product of the glycolysis and Krebs cycle, NADH and FADH2, this two. Okay. Let us see that the where we have got that in, again we can look back in glycolysis and the Krebs cycle, where all this were there. So, look at here the in glycolysis. In glycolysis, here is the place where 2 NADH plus H is produced. So, in between that glyceraldehyde 3 phosphate and 1,3 diphosphoglyceric acid. So, it will be 2 molecule. So, here only in this state, this first time that 2 molecule it started. Now, that is of the glycolysis. Glycolysis only the 2 molecule of NADH plus H is produced. Now, in Krebs cycle, the one before that one part we should discuss, that in between one part was there, that is in between glycolysis and Krebs cycle. Here also, here it is not there in the 2H, it means it is collected by NADH plus H. So, it is the one. And in cycle if you can see, these are here one NADH plus H, here one NADH plus H, here one NADH plus H, here FADH2. So, here inside in Krebs cycles, that total it comes 1, 2, 3, and in this, if you take that is the 4 and the 1. Okay. All right. So, total it comes in 10 NADH2 and 2 FADH2. 10 NADH plus H and the 2 FADH. Okay. If we can see this one, that will be the 10 and that will be the 2. All right. If you ask me that how can it possible, then I'm telling you from here, look at here, here one in a DH plus H, here it will be 2, because here the this Krebs cycle you have to multiply into 2 in every cases. So in between here also the same, here 2, here in any DHH, if you see that, that will be the 3, it means it will be 6, it will be 6. Now here one FADH2 is there, FADH2 in one cycle, but it will be 2. I'm reminding you that from the one glucose 2 molecule of pyruvic acid is produced. So when that pyruvic acid entered, we are calculating the 2 molecule. So it comes here total 10, here 2. So now you got that this is the 10 and this is the 2. Now we can enter inside the cycle. Electron transport system, it takes place inside mitochondria. You know this is the crystal, and here if we magnify, then what we get, that is the membrane of that crystal and that F1 particle, the oxisome, that F1 F0 particle. So here you can see that inside also this is, so this membrane, this is the membrane, here it is magnified, and this membrane here the electron will transfer and the proton is ejected on the another side. Next part we can see that this total electron transport is divided into 5 basic part. These are the complexes. Here we can see this the complex here, that complex 1, complex 2, complex 3, complex 4 and the 5. So let's know that in detail how the electron is transported. Okay, here you can see this part, this is the F1 particle, and this is the matrix. I think you can understand that inside mitochondria, and the inside crystal, that inner membrane, that space, this is the inner membrane space, this part is there. Okay. So from the matrix side 10 NADH plus H in complex 1 it will fix, and NADH dehydrogenase split this in flavin mononucleotide. Here it is, this one is all this here name, you can see that the acceptors of electron. So here it will be it will split into the proton and the electron, and the proton, all the proton is ejected in the intermembrane that space which is there, that is ejected, okay, from the 10 molecule. And this is complex 1. In complex 2, that FADH2 also that same, that is the in flavoprotein, it splits here in the proton here and the electron here. Electrons are it is carried. So here is a ubiquinone help, that it is called a mobile electron carrier, and it is carrying the electron. Okay. So proton remains another side, that intermembrane space, and the electrons are carried through the cytochrome B, cytochrome C1, cytochrome C. These are the mobile carrier, and ultimately it will again carried here, carried by cytochrome A1, cytochrome A3. So these are the carrier, and after that it is all this electron, it is accumulated in these areas. Look at here, it is divided into complex complex 1, 2, 3, 4. So, and all the proton here, it is separated. So now you can see this part, the proton here, all the proton in one sides, and the electrons are the another sides. So here is this situation also you have seen in the chloroplast when that energy is formed. Here also in this, which is called the chemiosmosis. So let's know a little bit about this one. So chemiosmosis, it is an energy coupling mechanism. In these that to move ions, that is a proton, to the other side of the membrane, resulting the generation of the electrochemical gradient and subsequent return ions downhill movement with the help of the membrane protein and some enzymes also. All right. So here it is proton gradient. In proton gradient, that energy is produced. So if you can see this proton, this proton here when it is accumulated, and here the ATPase enzyme, that is called the coupling factor. This huge proton storm will be there, huge storms, and here the electron it is there. So here the all the ADP, it turns into ATP. Look at here, the generation of energy. So here the energy is generated, and at the same time that oxygen which unites with hydrogen, here the water, and it neutralizes this. So this is the process of the energy generation. The first part is mobile electron carrier, electron transport, and chemiosmosis, where that proton gradient in the process, and I mean from the higher level of the proton, it goes to the electron, and this storm, the proton storm, will produce the energy ADP. With the help of the enzyme of ATPase enzyme, it converts into ATP. Now we can calculate that the amount of ATP which is formed. So how much energy yield from one molecule of glucose? That is, it is there, that is 38 molecule of glucose. Okay, 38 or 36 also, 36, yeah, 38 also in some books you can see that. In glycolysis, there are 4 molecule of the ATP produced, but 2 utilized. So there are gain here the 2. Here 2 NADH is produced. Each NADH will produce here 2 ATP. So here it is 4. After that, then in between stage, there is also 2 NADH, but here it is 6. Look at here, from 1 NADH here 3 molecule of ATP is produced, but here it is 2 molecule of ATP. This controversy is there still. Krebs cycles, the direct ATP is produced, and the 6 NADH it is into 3, 18, and FADH is giving 2 ATPs in per FADH2, so it is 4. Total it comes 36. If you consider this all NADH is 3, then it will be 38. So because here also will be 3, then it will be 6, then it will be 38. So 36 to 38, that energy it is produced. So that is about electron transport system. Next part we will do. Thank you.